Epigenetic regulation of thermogenesis.
The contemporary pandemics of obesity and diabetes are devastating in size and has been largely attributed to an environment which facilitates the consumption of junk food and sedentary behavior. Obesity can lead to insulin resistance, which is known as type 2 diabetes in previously unaffected population. Obesity develops when energy intake exceeds energy expenditure.
There are 3 types of adipose tissue. The white type is in charge of storing triglycerides as an energy source. The brown type burns glucose and lipids to dissipate energy by producing heat and maintain body temperature, thanks to an inner mitochondrial membrane protein called uncoupling protein 1. The beige adipose tissue cells express UCP1 under certain conditions. This is called browning of what? The thermogenic capacity of BAT makes it an attractive therapeutic target for inducing weight loss through energy expenditure.
BAT induction in mice promotes energy expenditure, reduces adiposity, and protects mice from diet-induced obesity. BAT ablation reduces energy expenditure and increases obesity in response to high-fat diets.
Now we are going to explain the epigenetic modification for activation of thermogenic genes. DNA methylation has a role in precursor cells to the adipocyte lineage, so the global methylation profiles of white and brown adipocyte precursors show it a distinguishable methylation pattern. In humans, DNA methylation at a specific lossy of markers for low or high response to calorie restriction. It is also important in the play between gene expression and environmental factors that may affect DNA methylation in brown and beige adipocytes. For example, an increased DNA methylation in addition to decreased histone acetylation correlates with the repression of thermogenic genes in the bud of hibernate rodents, limiting energy expenditure.
Through adding or removing numerous permissive or repressive histone marks, chromatin structure and gene expression are regulated during the differentiation of cells like adipocytes. These histone marks consist of methylations and acetylations, which are usually added in the N-terminal toll of histone 3. When a gene has the marks which are green, this expression is activated. On the other hand, red marks lead to its repression.
As we know, BAT has positive effects on glucose and lipid homeostasis and body weight. As a consequence, our interest is the epigenetic regulation that allows the expression of BAT genes. For example, the methyltransferase MLL4 is a writer responsible for adding this methylation mark on histone 3. If we add this mark to BAT genes, we increase their expression. On the other hand, the methylase LSD1 is an eraser that removes the same histone mark from white genes so that their expression is silenced.
Histone alterations are controlled at various points, such as expression levels of chromatin modifiers and this post-translational modification, affecting their activity and interaction with specific protein. Cold exposures and beta-adrenergic signaling are essential in the induction of these modifiers, thus promoting thermogenesis. An additional control level are specific metabolite concentrations, which regulate histone-modifying enzymes, acting as co-substrates and cofactors. Those metabolites are mainly acetyl-CoA, FAD, and NAD+, which levels reflect the metabolic status of cell, and by their epigenomic effect, they contribute to adaptation to the environment.